Hello and welcome, dear students. Today we are going to discuss about the topic human population genetics and forensics. The main objectives of today's lecture are to know the basics of human population genetics, to discuss about the origins and affinities of specific human populations, to discuss in detail about forensic genetics, to discuss the evolution of forensic genetics from blood groups to DNA profiles, to discuss the forensic significance of intragroup and intergroup variation, and finally to know the significance of human population genetics. Dear students, let's start with the introduction. The principles of population genetics attempt to explain the genetic diversity in present populations and the change in allele and genotype frequencies over time. Population genetic studies facilitate the identification of allele associated with disease risk and provide insight into the effect of medical intervention on the population frequency of a disease. Allele and genotype frequencies depend on factors such as mating patterns, population size and distribution, mutation, migration, and selection. By making specific assumptions about these factors, the Hardy-Weinberg law, a fundamental principle of population genetics, provides a model for calculating genotype frequencies from allele frequencies for a random mating population in equilibrium. Evolutionary forces such as random genetic drift, mutation, selection, and migration change the allele frequencies in a population. Important examples of each of these forces have been documented in human populations, and their effects are becoming better understood as knowledge of the genetic structure of populations at the DNA level increases. Mutations are the source of variation in a population and lead to change in allele frequencies and increased heterozygosity. The effect of selection on allele frequencies depends on the relative fitness of the genotype. In general, selection will lead to the eventual loss of an allele, but a large number of generations of selection may be required. However, if selection favors the heterozygote over both homozygotes, equilibrium allele frequencies will be reached. Ethnic variation in allele frequencies is found throughout the genome, and by examining this genetic diversity, evolutionary patterns can be inferred and variants contributing to the cause of common disease can be identified. As a result of major international initiatives, most recently the 1,000 Genomes Project, nearly 20 million single nucleotide polymorphisms or SNPs and copy number variants, that is CNVs, have been generated and catalogued. These extensive data sets provide the population geneticists with a huge set of densely mapped polymorphisms for reconciling genome variation with population histories of bottlenecks, admixture, and migration, and for revealing evidence of natural selection. The availability of massive databases of genetic variation and automated technology for genotyping, sequencing, and bioinformatic analysis is significantly enhancing collaborative efforts between population geneticists and molecular geneticists and advancing the understanding of many diseases. As we would anticipate, thousands of interesting new questions are arising, and the tools to answer many of them are now at our fingertips. Forensic genetics represents a combination of molecular and population genetics. Personal identification and kinship analysis, like that of the paternity testing, are the two main subjects of forensic DNA analysis. Biological specimens from which DNA is isolated are blood, semen, saliva, tissues, bones, teeth, hairs. Deoxyribonucleic acid, that is DNA, plays a vital role in forensic studies. Dear students, let's now discuss about the origins and affinities of specific human populations. Genetic data have been used extensively to study the origins and affinities of specific population groups. For example, analysis of genetic variation in Ashkenazi Jewish populations show that these populations are most similar to one another and to other populations from the Mid East and are distinct from non-Jewish European populations, although they have received some genetic contributions from these populations. Analysis of Y chromosome, mitochondrial DNA, and autosomal DNA support the relatively recent derivation of Native American populations from populations that lived in Northeast Asia. These data also indicate that there may have been substantial bottlenecks as the founding populations entered the Americas some 20,000 years ago. Another even more recent event is the human settlement of the Polynesian South Pacific Islands, estimated to have taken place just 3,000 years ago. Most evidence supports a Southeast Asian origin of Polynesian populations with mixtures between these individuals and Melanesian populations as they spread through the South Pacific. An analysis of mitochondrial DNA, Y chromosome, and autosomal variation in South Indian caste populations indicated less differentiation among castes for mitochondrial DNA than for Y chromosome DNA. This reflects greater levels of female, that is mitochondrial DNA gene flow among castes than male, that is Y chromosome gene flow. Dear students, let's now discuss in detail about forensic genetics. During the past 15 years, a revolution has taken place within the field of forensic genetics. Today, serious crimes are often solved by DNA investigations in which DNA profiles obtained from biological traces such as semen stains from the scene of a crime match the DNA profile of a suspect or a person in a criminal offender DNA database. In crime case work today, there is an international consensus to investigate repetitive DNA sequences in microsatellite regions, also called as short tandem repeats or STRs. Investigations are usually performed with enzymatic amplification of STRs, that is short tandem repeat regions, using the highly accurate technique of polymerase chain reaction or PCR, and analysis of the lengths of the STR region with a DNA sequencer. The DNA region that are examined are situated between the coding regions in the DNA, and the STRs do not code for any known characteristics. The polymorphism of an STR region mainly results from differences in the number of repeated sequences. This leads to variations in the total lengths of the STR regions from person to person. Very small amounts of stain material can be used because millions or billions of copies of the relevant DNA fragments of stain material DNA can be obtained by the PCR method. But a number of precautions must be taken during sampling and performance of the PCR-based DNA analysis to prevent contamination with irrelevant DNA. Dear students, a complete DNA investigation is a very effective tool for excluding individuals who are not very closely related to the person from whom the stain material was taken. A match between complete DNA profiles of a stain and a person is very strong evidence for the assumption that the stain came from that person compared with the assumption that the stain came from a random person. In paternity cases, most laboratories investigate several STRs with PCR-based methods. STR-DNA regions are inherited as classical Mendelian characters, and conventional genetic analysis of the results is done. Based on the knowledge of the distribution of the genetic markers investigated, the weight of the evidence for and against paternity is calculated. DNA typing of variable number of tandem repeat regions performed with the restriction fragment length polymorphism technique is done in some laboratories. In forensic genetics, the stimulus to investigate still smaller amounts of evidentiary material is closely associated with an increasing risk of errors attributable to contamination with DNA from other sources. In cases in which biological traces exist with mixtures of a relatively large amount of female DNA and a relatively small amount of male DNA, it can be difficult to obtain a conventional DNA profile of the material from the man. In such cases, it might be helpful to investigate STR regions on the Y chromosome because female DNA does not interfere with the typing of the male Y chromosome STR. Many single nucleotide polymorphisms or SNPs have been discovered in the human genome. For most human SNPs, only 2 DNA base variants have been observed, but SNPs with 3 or 4 allele exists. SNPs are less informative in forensic genetics than STRs and VNTRs, which are much more polymorphic, and a large number of SNPs are needed to obtain the same level of information as obtained by STRs. By using 50 to 100 selected autosomal SNPs, it should be possible in the future to obtain the same investigational results as those obtained with 10 commonly used STR systems. SNP typing can be done on small fragments of DNA, usually 40 to 50 base pairs. This fact is important in crime and identification cases in which DNA is severely degraded, perhaps as a result of fire or buried human remains. When traces of biological material from an unknown person are found at crime scene, information about physical traits such as color of hair, eyes, skin can be obtained. In the DNA region of the melanocortin 1 receptor gene, SNP factors associated with red hair and to some extent with fair hair and freckled skin exist. SNP markers in the DNA region coding for the agouti signal protein may be associated with brown eyes and black hair. The associations are weak with odds ratios of 1 to 10, and the investigations have only limited weight. The implementation of DNA investigation was the single most important event in forensic medicine in the late 20th century. The implementation and effective use of DNA databases are now the most important forensic genetic contribution to the fight against crime. Further, information gleaned from current and future human genome research projects will provide additional methods of solving crimes. Dear students, let's now discuss about the utility of genetic markers in forensics. Like other subjects, forensic genetics gradually emerged and developed through long-term social practice. Following the discovery of the ABO blood groups by Carl Landsteiner in 1901, human blood typing was used in identification, and forensic genetics entered the scientific age. In 1910, the French criminologist Edmond Locard proposed the Locard's exchange principle and stated that every contact leaves a trace, which laid the foundation for modern forensic science. In 1926, Thomas Hunt Morgan proposed a theory of genes which provided the basis for the development of forensic genetics. In 1953, the discovery of the double helical structure of DNA by Watson and Crick enabled the start of forensic genetics research at the molecular level. One of the central aspects of forensic genetics is the use of genetic markers, which are the easily identifiable phenotypes of genotypes. Genetic markers generally have features such as strong polymorphisms, codominant expression, and ease of observation and recording. With the development of genetics, the use of genetic markers has also developed steadily. To date, the development of genetic markers has gone through four major stages, that is characterized by the use of morphological markers, cytological markers, biochemical markers, and molecular markers. During the early 1990s, short tandem repeats were used for first time as key molecular marker in human paternity testing. STR typing based on capillary electrophoresis can only reflect differences in length. This provides a new possible method for difficult cases involving complex kinship identification and the resolution of mixtures. Since forensic polymorphic STR loci are limited in number and cannot provide more functional information, this hampers the effectiveness of the application in forensic community. Dear students, let's now talk about the role of SNPs in forensics. A new type of genetic marker, single nucleotide polymorphism, may replace STRs in the future, although this process may take a considerable period of time. The amplification products of individual SNP sites can be very short, which makes SNPs suitable for the analysis of highly degraded forensic samples. SNP analysis already plays an important role in identification of individuals after mass disasters. Furthermore, SNPs are linked with multiple phenotypes such as individuals' skin color, scleria color, hair curliness, information concerning ethnic origins, differences in alcohol metabolism, and susceptibility to hereditary diseases. These advantages of SNPs have all been successfully applied, and SNP analysis occupies an unsurpassed position in forensic practice. SNPs have historically been used to match a forensic DNA sample to a suspect, but has been made obsolete due to advancing STR-based DNA fingerprinting techniques. However, the development of next generation sequencing, that is NGS technology, may allow for more opportunities for the use of SNPs in phenotypic clues such as ethnicity, hair color, and eye color, with a good probability of a match. This can additionally be applied to increase the accuracy of facial reconstructions by providing information that may otherwise be unknown, and this information can be used to help identify suspects even without a STR DNA profile match. Some cons to using SNPs versus STRs is that SNPs yield less information than STRs, and therefore more SNPs are needed for analysis before a profile of a suspect is able to be created. Additionally, SNPs heavily rely on the presence of a database for comparative analysis of samples. However, in instances with degraded or small volume samples, SNP techniques are an excellent alternative to STR methods. SNPs, as opposed to STRs, have an abundance of potential markers, can be fully automated, and a possible reduction of required fragment length to less than 100 base pairs. Dear students, let's now discuss about the evolution of forensic genetics from blood groups to the DNA profiles. The very first known human genetic polymorphism variation in the ABO blood group system was identified by Carl Lindsteiner in 1900. From the outset, its potential in analyzing blood samples from crime scenes was recognized. A system such as this has low variability, that is few alleles and high average allele frequencies, so its power to attribute a biological sample to a particular person is correspondingly low. Samples from different individuals will often have matching blood types. By contrast, a non-match is strong evidence that can exclude a suspect at just this single locus as recognized by Landsteiner to detect the non-identity of blood samples. Forensic biologists went on to combine sets of these classical polymorphisms to reduce the random match probability. The chance that two different individuals have matching genotypes and exploited their Mendelian inherited nature in kinship testing. If genetic loci are unlinked and the population is randomly mating, then independent inheritance means that in principle their allele frequencies can be multiplied in deriving genotype frequencies. This is known as the product rule. As a consequence, RMPs fell to more useful average levels of 1% or lower, but there remained practical problems of protein degradation, body fluid specificity, and the interpretation of mixed samples. It was the development of DNA-based analysis in the mid-1980s that relegated Levotin's classical polymorphism from forensics and began the modern era of robust individual identification. Initially, this was via DNA fingerprinting based on length variation at multi-allelic autosomal mini-satellites, and by the 1990s, DNA profiling based on length variation at short tandem repeats, that is STRs, also known as microsatellites. Dear students, let's now understand the forensic significance of intragroup and intergroup variation. The forensic genetist needs genotypes that provide robust individual identification and aim that emphasizes variation within the population. Calculating an RMP to evaluate the significance of a match can then be done by compiling allele frequencies, data from that population, and assuming homogeneity and random mating. When a crime scene profile matches a suspect, but the only information available about the perpetrator is the DNA profile itself, then the choice of reference population for calculating the RMP becomes particularly important. If there were major differences in allele frequencies between different populations, then a DNA profile might be very rare in one population, strongly incriminating the suspect, but orders of magnitude more common in another. Declaring the end of the DNA fingerprinting wars without solving the underlying issues means that questions of interpopulation variation and population substructure have not disappeared and tend to arise afresh with each new development in forensic technology. In today's age of highly sensitive PCR multiplexes, STR profiles are often partial, missing a full set of loci or alleles, or mixed, which can increase RMPs and make interpretation more challenging. More rigorous approaches to calculating RMPs under different models of mate choice have been developed, as well as considering the significance of a match between the profile of a known suspect and a crime scene sample. In the modern world of very large investigative databases such as those of the US and the UK, cold hits are often reported and evaluated. Dear students, let's now know about the significance of human population genetics. Population genetics, genomics, is used for detecting genetic diseases and genetic risk factors for multifactorial disease, understanding diseases using insights obtained from genetic risk factors, and treating disease using these insights. Population genetics also underlies the field of evolutionary medicine, understanding and treating some of our present diseases in terms of the environments under which they evolved. A final issue is whether or not humans are no longer evolving because cultural evolution has replaced biological evolution. Quite the contrary, cultural evolution induces strong selective forces, so humans have undergone rapid adaptive evolution over the past 10,000 years. Human population structure is also rapidly changing, with dispersal and outbreeding becoming much more common, coupled with major demographic transitions and population growth. These changes affect both adaptive and neutral evolution. One possibility for human evolutionary change is eugenics, the deliberate modification of human reproduction or specific genes to change our gene pool. Eugenic proposals are most often based on genetic determinism that ignores the fact that phenotypes emerge from interactions between genotypes and environments. Eugenic proposals also tend to ignore the role of epistasis and pleiotropy, which together seriously undermine the eugenic agenda. Humans have great potential to continue to evolve both biologically and culturally in our rapidly changing environment. Dear students, let's now conclude the lecture. Population genetics is the branch of genetics dealing with the description of observed or inferred heritable features in populations through space and time. Furthermore, population genetics can be dealt with in purely theoretical terms or in a wide range of applications, such as in forensics. Population genetics is the science of genetic variation within populations of organisms. Population genetics focuses on the origin, amount, frequency, distribution in space and time, and the phenotypic significance of that genetic variation, and with the microevolutionary forces that influence the fate of genetic variation in reproducing populations. The extensive theoretical evidence of this field stems from three premises: DNA can replicate, DNA mutates, and it recombines. And the information in DNA interacts with the environment to produce the traits or characters. DNA replication means that a single type of gene can exist in space and time across generations beyond any individual who bears the gene. The fundamental importance of population genetics is the basic insight it provides into the mechanism of evolution. It would be unimaginable that forensic scientists will give up their efforts to maximize intelligence from DNA evidence. However, it is also important to remember that these creative endeavors are undertaken because of the absence of universal forensic DNA databases. Indeed, the biases, ethical problems, and invasions of privacy that the armory of investigative methods present have been used to bolster the arguments for universal databasing. There is a clear need for good practice in considering human classifications in imperfect but important forensic probability estimates. Labels matter and should be used more carefully. This should include a nuanced consideration of admixture, rather than the shoehorning of DNA donors into individual groups. It is promising that the field has woken up recently to the issue of ethics, and in particular to the question of informed consent of participants in forensic population studies. This suggests that the broader questions around how forensic genetics interacts with racial classifications and a public view of human differences should also be the subject of consideration and regular re-evaluation, rather than relying on tablets of stones from a previous era representing empirical and arbitrary standards. Dear students, it was all about today's lecture regarding human population genetics and forensics. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.